Assalamualaikum and hi everyone. In this video, we are going to discuss on chapter 6, expression of biological information, subtopic 6.2, DNA replications. In this subtopic, we have a few learning outcomes that we need to be achieved. However, I'm going to specifically focus on the second learning outcome, explain the enzyme and protein involved in DNA replication. Meanwhile, for learning outcome 6.2a, it already discussed on previous video. And for learning outcome 6.2c, it will discuss in your tutorial class. Okay, they have 5 enzymes and 1 protein that involved in DNA replication, which is DNA helicase, single-strand binding protein, topoisomerase, primase, DNA polymerase, which is involve the DNA polymerase 3 and DNA polymerase 1, and the last one is DNA ligase. The first enzyme involved in DNA replication is DNA helicase. The function of DNA helicase is to unwind the double-stranded DNA to become single-stranded DNA. So if we look at this diagram, so this is a double-stranded DNA, parental DNA, and this one, the purple in color, is the helicase. So helicase will unwind the parental double helix, so from the double-stranded DNA to become single-stranded DNA. The protein involved in DNA replication is single-strand binding protein. The function of single-strand binding protein is to stabilize and prevent the single-stranded DNA from recoiling in helix until the strands are replicated. So if you look at here, so single-strand binding protein bind to the unpaired DNA strand, keeping them from repairing after the helicase unwind the parental double helix. The second enzyme is topoisomerase. It helps relieve the strain caused by the unwinding of DNA double-stranded. So this one is the helicase. When the helicase unwind the DNA double helix, it will cause tighter twisting and strain ahead of the replication fork. So that we need the topoisomerase to relieve the strain. The third enzyme is primase. The function of primase is to synthesize RNA primer by adding RNA nucleotide that complementary to DNA template. If we look at this diagram, the blue in color is the single-stranded region of parental DNA, or we call it as the DNA template. The green in color is the primase. So the primase join RNA nucleotide into primer. So the pink in color is the primer. The elongation of primer is from 5' end to the 3' end. The fourth enzyme involved in DNA replication is DNA polymerase. So we have DNA polymerase 3, which is function to add DNA nucleotide to the 3' end of the short RNA primer. So the pink in color is the RNA primer. So we have 5' end to the 3' end of RNA primer. So the newly made of DNA is added to the 3' end of short RNA primer by the DNA polymerase 3. Next is DNA polymerase 1. The function of DNA polymerase 1 is to replace the RNA primer by adding free DNA nucleotide, producing new complete DNA strand. So the previous one, we are talk about the DNA polymerase 3 added the free nucleotide in the to the 3' end of the primer. So the primer is come from the RNA nucleotide. So the process of DNA replication is we want to replicate the DNA. Okay, to make the new complete of DNA strand, we need to remove the RNA primer. So by using the DNA polymerase 1, it will replace the RNA primer. The last enzyme involved in DNA replication is DNA ligase. The function of DNA ligase is to form phosphodiester bond to join Okazaki fragment together at lagging strand. As we discussed earlier, the function of DNA polymerase 1 is to replace RNA primer with the free DNA nucleotide. But the DNA polymerase 1 cannot join the final nucleotide of this replacement DNA fragment to the first DNA nucleotide of the adjacent Okazaki fragment. So that's why we need DNA ligase to form phosphodiester bond to join the Okazaki fragment. Okay, so we repeat again the 5 enzymes and 1 protein that involved in the DNA replication. So we have the DNA helicase here, and then the second one is the single strand binding protein. Okay, and then we have topoisomerase to relieve the strand, and then we have the primase. Okay, to add the RNA primer, and then we have DNA polymerase, which is we have DNA polymerase 3 and DNA polymerase 1, and the last one we have the DNA ligase. So that's all for enzyme and protein that involved in the DNA replication. And as a quick check, so please answer the question. Thank you.